Now, macromolecules that will find in the acid insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides. Together with the polysaccharide and polypeptides, these are made up of the true macromolecule fraction in any living tissue or cell. For nucleic acids, the building block is a nucleotide. The building block is a nucleotide, a simple unit. And a nucleotide has three chemically distinct components. One is a heterocyclic compound. The second is a monosaccharide. And the third is a phosphate acid or phosphate group. So we see that the heterocyclic compounds in nucleic acids are the nitrogenous bases. Nitrogenous bases, these can be adenine, thymine, cytosine, uracil, etc. Adenine and guanine, they are substituted purines, while the rest they are pyrimidines. Okay, purines they are one ring and pyrimidines they are double ringed. So the skeletal heterocyclic ring is called as a purine and a pyrimidine respectively, and the sugar found is a polynucleotide which can be either ribose, that is a monosaccharide pentose, or a 2-deoxyribose. So the nucleic acid containing deoxyribose is called as the DNA, deoxyribonucleic acid, while that which contains a ribose sugar is called as the ribonucleic acid or RNA. Now the structure of proteins. Proteins, they are heteropolymers and they contain strings of amino acids. They have strings of amino acids, like you can see here, different amino acids, they are combined together. So in inorganic chemistry, the structure refers to the molecular formula with the NaCl, magnesium chloride, and organic chemists they write the two-dimensional view of the molecules, which represents the structure of the molecule like benzene, naphthalene, etc. So physicists they have a three-dimensional view of the molecular structure, while biologists they describe the protein structure at 4 levels. The sequence of amino acids, the position of the information of a protein, which is the first amino acids, which is second, and so on. And they call it as the primary structure. So this structure is called as the primary structure of the protein. So the protein is imagined as a line. So it is imagined as a line, and which represents the first, and it is the left end represents the first amino acids and the right end represents the last amino acids, any acid. So the first amino acid is also called as the N terminal. It's also called as the N terminal, like you can see here, and the last amino acid is called as the C terminal. It is also called as the C terminal. So a protein thread, it does not exist without an extended rigid rod. So the thread is folded in the form of a helical structure, which is similar to a revolving staircase, and only some portion of the protein thread they are arranged in the form of a helix. So in proteins, only right-handed helices they are observed. So in other regions of the protein, the threads are folded into other forms in which they are called as the secondary structures. You can see here a secondary structure. Secondary structure, the helix are like this, and it's a tertiary structure of a protein. So the long protein chain is also folded upon itself like a hollow woolen ball. So that gives it a tertiary structure. So this gives us a three-dimensional view of the protein. So tertiary structure is necessary for many biological activities of the proteins. Some proteins are an assembly of more than one polypeptide or subunit. So the manner in which these individual folded polypeptides or subunits are arranged with each other, the linear strings, spheres arranged upon each other in the form of a cube or a plate, so that is called as a quaternary structure of a protein. So in the human hemoglobin, it contains 4 subunits and 2 of these are identical to each other. So the 2 subunits of alpha type of the 2 subunits of the beta type together, they make the hemoglobin, human hemoglobin. Now, nature of bond linking monomers in a polymer. Polypeptide or a protein, amino acids, they are linked by the peptide bond. You can see here, these are the peptide bonds where the two amino acids, they are linked together. So when a carboxyl group, you can see here the carboxyl group of one amino acid, it reacts with the amino group of the second amino acid with the elimination of a water molecule. When a water molecule is eliminated or removed, and this process is called as dehydration. So whereas in polysaccharides, we see that the individual monomer units are linked by glycosidic bond. Monomer units, they are linked by the glycosidic bond, and the bond is also formed by dehydration. Dehydration means removal of a water molecule. So this bond is formed between two carbon atoms of the two adjacent monosaccharides. So in a nucleic acid, a phosphate links the 3 carbon of one sugar and one nucleotide of 5 carbon to the sugar of the other nucleotide. And the bond between the phosphate and the hydroxyl group of sugar is an ester bond. And as there is one such ester bond on either side, it is called as the phosphodiester bond also. You can see here, this is the glycosidic bond, which is formed by the individual monosaccharide units when they are linked together. And here you can see the structure of the DNA. Nucleic acids, they have a secondary structures. One of the secondary structures of the DNA is the famous Watson and Crick model. This model says that DNA exists as a double helix. The two strands of polynucleotides, they are anti-parallel, that is run in the opposite direction, and the backbone is formed by the sugar and phosphate. So this backbone is formed by the sugar and phosphate groups. And the nitrogen bases, they are projected perpendicular to the backbone, but faces, like adenine faces the thymine, and the cytosine to the guanine. So we see there are 2 hydrogen bonds between adenine and thymine, and 3 hydrogen bonds. There are 3 hydrogen bonds between cytosine and guanine. So each strand appears like a helical staircase, and each step of the this is called as the base of the staircase. And we can see here the line diagram. And the pitch would be here the pitch would be 34 angstrom, and the rise per base pair would be 3.4 angstrom. So this forms the DNA with the salient features of the DNA, and we will be studying about the structure in detail in the higher classes. Now, dynamic state of body constituents, or the concept of metabolism. Living organisms like a simple bacteria, protozoa, plant or animal, they have thousands of organic compounds. Now these compounds are biomolecules, and they are present in certain concentrations which are moles per cell or moles per liter. Now these biomolecules, they also these biomolecules they are also we observe that they are keep changing all the time into some other biomolecules, and also they are made from different biomolecules. So this breaking and making through different chemical reactions is going on in the living organism. So all these breaking and making processes, that is called as the metabolism. So each of the metabolic reactions results in the formation or transformation of a biomolecule. So here metabolic transformations, that is removal of carbon dioxide from amino acids, and making amino acid into an amine, removal of amino group from a nucleotide base. From a nucleotide base, if we remove the amino group, so that will result in the making of, if we remove the amino group from the amino acid, so it will make it an amine. Such different types of reactions they are going on. Hydrolysis of glycosidic bond in disaccharide. So there are many such reactions. They do not occur in isolation. All these reactions, they are always linked to each other. So the metabolites, they are converted into each other through a series of linked reactions which are called as metabolic pathways. So this metabolite flow, that is dynamic state of body constituents, they are all interlinked. And another feature of these metabolic reactions, that even chemical reaction is catalyzed reaction. Catalyzed reaction means catalyst which speeds up a biochemical reaction. They are also there. So there are no uncatalyzed metabolic conversions in living system. Even carbon dioxide, which is dissolving in water, a physical process, is a catalyzed reaction in the living system, and the catalyst which increases the rate of a given metabolic conversions, they are also proteins. So these proteins with catalytic power, which they are called as enzymes. Metabolic basis for living and the living state. Metabolic pathways, they results in a more complex structure from a simple structure. For example, acetic acid becomes cholesterol, and a simple structure from a complex structure, for example, glucose becomes lactic acid in our skeletal system. So these are anabolic and catabolic pathways. Anabolic pathways, they consume energy. Anabolic pathways, they consume energy, and proteins from amino acids, they also require energy. Catabolic pathways, they results in the release of energy when glucose is degraded to lactic acid. So when glucose is degraded to lactic acid, it is in our skeletal muscles, so energy is liberated. This metabolic pathway from glucose to lactic acid, which occurs in 10 metabolic steps, is called as glycolysis. So this one step reaction is actually a 10-step reaction in the form of a glycolysis. Living organisms, they trap this energy during this process, and it is in the form of chemical bonds, and as and when needed, these bond energy is utilized for biosynthetic, osmotic and mechanical work. Now all this energy is in the form of ATP, adenosine triphosphate. So during anabolism, the complex molecules, anabolism is a synthesis process where the complex molecules are formed with the help of the energy, whereas opposite is the catabolism, where the complex molecules are broken down into simpler ones with the release of energy. The living state. Now living organisms, they work continuously and they cannot afford to reach an equilibrium. If they reach an equilibrium, then their state will become stable. So living state is a non-equilibrium state which is able to perform work, and living process are the constant effort to prevent the equilibrium. If they'll come to an equilibrium, then there'll be a steady state. There is no change happening. So whenever they are in a non-equilibrium steady state, then they are able to perform the work. So for this they require energy input. So metabolism is a process which provides this energy. So the living state and metabolism, they both are synonyms, and because the living state means metabolic reactions are going on, and without metabolism, living state cannot be achieved.